All right, so I just want to make some concluding remarks. As is always the case in biology, we have exceptions to the central dogma. So I mentioned that the central dogma describes the direction of the flow of information from DNA to RNA to protein, but there are examples of like retroviruses and retrotransposons. They actually have RNA that they can reverse. They use reverse transcriptase to create a double-stranded DNA, which can be then inserted into the host genome, then replicated along with the host genome, and then transcribed into RNA. And so that's one example where the central dogma does not stand, at least the directionality. And then another really cool area of research is non-coding genes. So coding genes mean that they encode for codons, right, amino acids. Non-coding genes do not code for amino acids. So rather they create these non-coding RNAs that are transcribed, but then they're not ultimately translated. And they're used in different fine-tuning of gene expression and regulation of gene expression at like non-protein levels. And then I just want to make a final comment, because this is kind of the like pre-video for a lot of really cool functional genomics videos that are coming in the future. Gene regulation goes beyond the central dogma. So here I was just talking very specifically about genes and how they are expressed. How do we get the gene information from inside the nucleus at the DNA level out into the cytoplasm to create proteins so that we can have cell type specific functions and create, a full organism. But there's a lot more to our DNA. The coding genes are only 1% of the DNA, and we only have about 20,000 of them, but way, way, way more functions, that like more than you can imagine. So it's kind of exciting, and yeah, stay tuned and keep watching. And I just want to thank again our sponsor, UCLA Computational Medicine, for the video setup and the space, and for hosting our videos on the Big Bio website. Thank you. Yeah.